We strongly encourage users to follow along with our written protocol located on our website at the link displayed. Before utilizing the microCT 50, ensure that you have all the necessary materials to conduct your experiment. First, begin by pipetting a warm layer of agar onto the petri dish to create a base layer. Allow 5 to 10 seconds for this base layer to cool. Before placing your first stack of bones, we strongly encourage users to record any sample information on our dedicated scanning parameters document, which is located on the Musculoskeletal Research Center's website under Corby's Technical Notes and Presentations tab. This link is also provided in the video description below. Place your first stack of bones onto the base layer of agar, ensuring they fit within the boundaries, are all in an identical orientation, and are as close to each other as possible. Record each bone's position on the previously mentioned scanning parameters document. Once your first stack of bones has been set, pipette a thin layer of agar on top of the first stack of bones and allow it to cool for 5 to 10 seconds before placing your second stack of bones. If at any time throughout this process the agar begins to solidify, reheat it and ensure it is cool enough before using it again. While placing your second stack of bones, as mentioned with the first stack, please be sure to keep them in a vertical orientation and as close to each other as possible. After the bones have been set in their respective positions, place the fiducial marker in the 6th position. As always, ensure this marker is vertical and as close to the bones as possible. Pipette an additional layer of agar on top of the second stack and allow 5 to 10 seconds for this layer to cool. Finally, pour a generous layer of agar into the petri dish until it's full. Allow this dish to cool for 5 minutes before proceeding. Check the firmness of the agar by first using the forceps and then using your hand. If the agar is firm, it is ready to cut. Carefully, using the razor blade, cut out the Agar-Bone block as shown. Ensure not to cut too close to the bones, as one can always trim the Agar-Bone block if necessary, but cannot add additional agar without restarting the process. Once you have finished cutting, safely store the razor blade and gently use the Weibo spatula to remove the Agar-Bone block from the petri dish. Be sure not to injure the Agar-Bone block throughout this process. It is strongly encouraged to remove the uncontaminated agar first to prevent damage to the Agar-Bone block. Once the Agar-Bone block has been removed, please be sure to return the uncontaminated agar back to the jar of agar. Before moving on to the next step, ensure a removal button is placed inside the specimen tube with the wires facing upward. When ready, place the Agar-Bone block inside the specimen tube with the bone's distal end first. If the Agar-Bone block cannot fit, trim the edges using a razor blade. Push the Agar-Bone block down the tube using a pushing rod and then firmly cap it, ensuring nothing is protruding outside of the specimen tube. Slowly open the microCT's hatch and begin placing your tubes on the carousel. Orient the flat side of the metal base so that it is in contact with the flat side of the dodecagon on top of the carousel. Be sure to record which tube is in which carousel position. Slowly close the hatch once all tubes are placed. Never load more than 11 samples or tubes in the microCT 50 at a time. The 12th slot is kept vacant for scanning emergencies only. After the microCT 50 has warmed up, click Yes on the message and wait for the CT to acknowledge and identify the tubes. Once prompted to do so, enter your sample number, which should have been provided by the lab manager. Firstly, clear the previous tasks by clicking the Task List button at the bottom of the screen. Next, click the Clear List button, followed by Yes to confirm. Firstly, check to ensure your sample number and name are correct. Next, search for your custom control file in the Filters box, which should have been provided by the lab manager. Select a carousel position at the top of the screen, select your control file, and then click Add Scout, as shown. Repeat this selection process for each tube. Once all of your tubes have been associated with a control file and have been added as pending tasks in the task list window, click the Start Interactive Task button to initiate the ScoutView scans. Once complete, you'll note that the ScoutView tasks will have converted from pending to done. To load a ScoutView, select a task, and then click Load Scout. To adjust the ScoutView, use the mouse wheel to zoom in or out, and the right-click button for any translational movements. Once satisfied, click the Reference Line button to create your reference markers for each scan. While hovering over the ScoutView, find the starting point and lock it in place by holding the Shift key. While still holding the Shift key, move the mouse to the ending point and left-click to set the reference marker in place. If a mistake is made, click the Reference Line button again and restart the marking process. Once satisfied, click the Add Scan button to create a pending CT scan task, which will appear as a new task in the Tasks window. In order from top to bottom, as shown, repeat this reference marking process for each task labeled as a ScoutView. Once all of your samples are converted into pending CT scan tasks, ensure one of the CT scan tasks is selected, then click Submit Batch Scans at the bottom of the window. This initiates the microCT 50's scanning process. A blue loading circle will appear for about 2 to 3 minutes before all SCANCO windows disappear. Once this disappearance occurs, it indicates the scan for the sample has begun. During the placement of the reference markers, an estimated time for that tube's scanning period is displayed in green text. Please add up these times for each tube and swiftly remove your tubes from the microCT 50 upon completion. Additionally, as always, we strongly encourage users to follow along with the written protocol, which is linked in the video description below. Before analyzing any data, we must first load the samples and subdivide the bones into independent measurements. Load the evaluation program and locate your evaluation group either from the drop-down menu or via filtration. Once you have located your evaluation group, load the first measurement by either double left-clicking on it or by selecting the .isq file and clicking Load. Next, navigate to the Tasks tab, which is located at the top middle of the screen, and then enter the Script drop-down menu as shown. Next, select a thumbnail slice, located on the right side of the screen, that displays all bones within the tube. Refer to the drawing you made of the bone's positions and adjust the volume of interest box around the bone in the first position. Ensure this box fits around the bone for all of the thumbnail slices. If not, click on the thumbnail slice where the box appears smaller and adjust as necessary. During this process, you are isolating and subvoluming each bone such that it can be placed in its own measurement, which, as you will see later in this tutorial, makes the analysis process more efficient. Before the subvolume takes place, double-check to ensure the VOI box fully encapsulates the bone of interest and allow for some padded room around the bone to prevent partial bone exclusion. Once satisfied with the VOI box's placement, navigate to the measurement naming box at the bottom left corner of the screen. Name this measurement with your PI's name, the bone's position, and any other pertinent information. You are now ready to start the subvolume script. locate and click the Start Task button to initiate the script. Click on the History button located just to the right to view the job queue history. Here you'll note the status of the subvolume script is pending, indicating the script is in a queue. The status will convert to running when the script initiates, and success when the script has finished. Unfortunately, we cannot subvolume the next bone until the script is finished running, as we would be adjusting the VOI box within the same measurement, causing the script to fail. Once the script is finished running, you may close out of the task history window and, in order, repeat the subvolume process for each bone within the measurement. Once you are finished with the subvolume process, at the top left corner of the screen, click Projects, then Scan Group Members. In this new window, search for your subvolumes via filtration. After locating your subvolumes, while holding the Control key on your keyboard, select all of them. Find your designated evaluation group, which should have been provided by the lab manager, and then click the Add To button in the bottom left corner. In this case, we'll be adding the test samples to the training evaluation group. Next, click the Refresh button in the top right to ensure your measurements have been moved. You may close out of this window once this is complete. Before beginning the next step, we strongly encourage users to download and print these analyses tables to help keep track of the analysis. These two images displayed represent the trabecular analysis conducted in this tutorial video. The analysis is performed over a 150 slice range from a starting slice of 1766 to an ending slice of 1916. To acquire both the analysis starting and ending slices, we must first locate the slice at which the growth plate begins. In this example, that slice value is 1916 and is otherwise known as the analysis end slice. Next, we can subtract 150 slices from the analysis end slice to acquire the analysis start slice. If you are experiencing any difficulties during the trabecular analysis or have questions regarding where to analyze, please do not hesitate to contact the lab manager for additional assistance. As an additional reminder, we strongly encourage users to follow the written standard operating procedure located at WashU's Musculoskeletal Research Center website under Core B's Technical Notes and Presentations tab. As shown during the Analyzing Your Scans chapter of this video, load your first measurement, which is usually the bone in the first position. Once loaded, select the thumbnail slice that appears as if it contains trabecular bone. Assuming you inserted the Agar-Bone block with the distal end first, this slice will be located near the end of the scan. Using the slider bar in the bottom right, attempt to locate where the growth plate begins. Use the left and right arrow keys on your keyboard to move one slice at a time as you near the epiphyseal plate. Once you have found the slice that the growth plate starts at, record this value as the metaphyseal start slice. Repeat this process for each measurement. Reload your first measurement as shown in the Analyzing Your Scans chapter of this video. First, navigate to the Tasks tab and then locate the script drop-down menu in the bottom left region. Click on script number 11, which is titled Auto Contour Bone. At the bottom left corner, replace the default Z value with your Trabecular Analysis start slice and alter the DZ value appropriately. In this example, we will replace the DZ value with 150 to create contours for 150 slices past the analysis start slice. Finally, ensure trabecular is selected from the drop-down shown. At the bottom right of the screen, input the Trabecular Analysis start slice in the slice position box and hit the Enter key on your keyboard to load the slice. Adjust the VOI box around the bone of interest in the same fashion as done during the subvolume process. We must check to ensure the VOI box fully encapsulates the bone of interest for the entirety of the 150 slices. To do so, use the 10 slice technique by clicking on the slider bar. Each click moves the slider 10 slices in that respective direction. Adjust the VOI box as necessary. Locate and click the Start Task button to initiate the auto contour script. As shown earlier, the history window will display the job queue history and the status of the script. Because the bones have been subdivided, they each reside in their own measurement, allowing us to load the next bone while the script is still running. Repeat this process for each measurement. After the contours have been created, we must check to ensure they have been drawn correctly. Reload the first measurement as shown in the 8th minute of this video. Once loaded, navigate to the Contouring tab and locate the array of green boxes or individual slices at the bottom middle of the screen. Select the first slice and then use the 10 slice technique to check the accuracy of the contours. Please contact the lab manager if the contours are drawn improperly. Repeat the process of checking the contours for each measurement. After the contours have been checked, please reload the first measurement, navigate to the Tasks tab, and select your custom Trabecular Analysis script from the Script drop-down menu. Next, locate the slice position box at the bottom right region of the screen and load a slice in the analysis range. If necessary, adjust the VOI box to fully encapsulate the bone. Use the Mask and Preview buttons as shown, or the F5 and F6 keys on your keyboard to switch between the mask and the preview. The goal is to match the mask's trabecular area with the trabecular bone. To do so, adjust the lower threshold value as necessary to alter the mask's area. After a good threshold value has been determined, quickly check if this value is appropriate for a few more slices in this measurement. If not, adjust the value accordingly. Finally, record this value for later use. Repeat this process for an additional 3 to 4 measurements within a sample group, such as a control group, knockout group, drug group, etc. Ensure to record all of these lower threshold values, as the mean or average of these values will yield the finalized lower threshold value to be used for the analysis script. Once a finalized lower threshold value has been calculated, we can begin the analysis process. Reload your first measurement as shown in the Analyzing Your Scans chapter of this video, navigate to the Tasks tab, and select your custom Trabecular Analysis script from the Script drop-down menu. In the bottom left corner, replace the current Z value with your Trabecular Analysis start slice and the DZ value appropriately. In this example, we'll be analyzing 150 slices. Before running the script, ensure the lower threshold value is correct. If not, adjust it accordingly. When ready, click the Start Task button and then click OK. As mentioned previously, because the bones have been subdivided into independent measurements, we can load the next measurement and continue the analysis. These two images displayed represent the cortical analysis conducted in this tutorial video. The analysis is performed over a 50 slice range from a starting slice of 1044 to an ending slice of 1094. To acquire the analysis starting and ending slices, we must first acquire the bone's starting and ending slices. Once the starting and ending slices of the bone have been found, find the midpoint between the two ends. In this example, the midpoint slice is 1069. Next, subtract 25 slices from the midpoint to acquire the analysis start slice, and add 25 slices to the midpoint to acquire the analysis end slice. If you are experiencing any difficulties during the cortical analysis or have questions regarding where to analyze, please do not hesitate to contact the lab manager for additional assistance. Additionally, feel free to return to this section of the video as needed. Again, we strongly encourage users to follow the written standard operating procedure located on WashU's Musculoskeletal Research Center website under Core B's Technical Notes and Presentations tab while watching this video. Before beginning the cortical analysis, please note that the following steps are primarily intended for femur bone analysis. Reload the first measurement as mentioned previously and navigate to the Tasks tab. Select the thumbnail slice closest to the proximal end of the bone, and then use the slider and the left and right arrow keys on your keyboard to locate the slice in which the first bit of bone appears. This is otherwise known as the starting slice. Record this value for later use. Next, select the thumbnail slice closest to the distal end of the bone, and use the slider again to locate the ending slice, or the last bit of bone that appears. Again, record this value down. After both slice values are determined, move on to the next measurement and repeat this process for each bone while ensuring these values have been recorded. We are now ready to determine the lower threshold value for the cortical bone. Reload your first measurement as mentioned in the 8th minute of this video. Next, navigate to the Tasks tab, locate the Script drop-down menu, and then select your custom Cortical Analysis script. In the bottom left corner of the screen, replace the Z value with your Cortical Analysis start slice and your DZ value appropriately. For this example, we'll be analyzing over a range of 51 slices. Next, locate the slice position box at the bottom right region of the screen and load a slice in the Cortical Analysis range. If necessary, adjust the VOI box to fully encapsulate the bone. Use the mask and preview buttons shown or the F5 and F6 keys on your keyboard to switch between the mask and the preview. The goal is to match the mask's cortical area with the cortical bone. To do so, adjust the lower threshold value as necessary to alter the mask's area. After a good threshold value has been determined, quickly check if this value is appropriate for a few more slices in the same measurement using the 10 slice technique. If not, adjust the value accordingly. Finally, record this value for later use. Repeat this process for an additional 3 to 4 measurements within a sample group such as a control group, knockout group, drug group, etc. It is not required to repeat this process for every bone or measurement. Ensure to record all of these lower threshold values, as the mean or average of these values will yield the finalized lower threshold value to be used for the analysis script. Once a finalized lower threshold value has been calculated, we can begin the analysis process. Reload your first measurement as shown in the Analyzing Your Scans chapter of this video. Navigate to the Tasks tab, and then select your custom Cortical Analysis script from the Script drop-down menu. In the bottom left-hand corner, replace the current Z value with your Cortical Analysis start slice and the DZ value appropriately. In this example, we'll be analyzing 51 slices. Before running the script, ensure the lower threshold value is correct. If not, adjust it accordingly. Return to the slice position box, load a slice in the Cortical Analysis range, and then adjust the VOI box to fully encapsulate the bone. Using the 10 slice technique, check to ensure the VOI box fits around the bone throughout the Cortical Analysis slice range. Finally, click the Start Task button, then click OK. As mentioned previously, because the bones are subdivided into independent measurements, we can load the next measurement and repeat the analysis process. To export your analyzed data, navigate to the Application Center, click Tools, and then click File Browser. Navigate to the Projects tab, and then select the Results folder. If you have a scan group, attempt to locate your respective folder here. If you cannot locate a scan group folder, check to see if your data is in the All folder, either as an evaluation group or mixed with other loose data. After locating your data, you may export it to the local desktop hard drive. To do so, right-click on your selected file and click Download. In this new window, select the Downloads folder and right-click in the open space to create a custom folder. Name this folder similarly to how your data was named. For this tutorial video, we have named our export folder as SOP Test. Double left-click on the folder to enter it, and then click Save at the bottom right of this window. This folder may also be used for your future data storage. To convert a SCANCO.isq file to a DICOM, first load the measurement you wish to convert. Navigate to the Tasks tab, click on the Script drop-down menu, and then select the script number 24 titled Convert to DICOM. In the bottom left corner, ensure the drop-down settings are identical as shown. The 8-digit scan number usually represents the 4-digit number located at the top of the SCANCO window. Memorize or record this value to help identify it when retrieving and exporting data. When ready, click the Start Task button and then click OK. If you have additional .isq files to convert, you may now load them. Remember, you may only load the next measurement while a script is running if the bones are independent measurements. To access and export your DICOM data, navigate to the Application Center, click Tools, and then click File Browser. In this new window, click on the Projects tab. Then, double left-click on the Exports folder. Using the scan number mentioned earlier and the modification dates, locate your DICOM file. Following the download process at the beginning of this section, store this file appropriately. For the purpose of this tutorial video, we'll be storing the DICOM data in our SOP test folder. Use the following steps if a script has already run on the image that is to be viewed as a 3D image. If not, skip to the next chapter. First, navigate to the Application Center and load the Ray Tracer window. Wait for the Select Sample and Measurement window to appear before selecting your sample. Next, select the All Files bubble at the bottom middle. A list of measurements will appear in the box on the right. Locate your respective measurement and the file extension that is currently shown. Click OK once you have selected a file. To load the 3D image, click the Start button at the bottom left of the window. Please wait for the loading circle to disappear before altering the image or making changes. We strongly encourage users to play around with the software to become more comfortable with its features. But remember to wait for the image to fully load before making the next change. If you wish to export your 3D image, skip the following chapter. Use the following steps if a script has not already run on the image that is to be viewed as a 3D image. First, load the image of interest. Once loaded, navigate to the Contouring tab, and then locate the Slice Position box in the bottom right corner of the screen. Enter a starting slice value for your 3D image and click the Enter key on your keyboard to load the slice. Next, in the bottom left corner, select the Ellipse or Circle tool to draw a contour around the bone of interest. Use the Select tool to adjust the contour's position and its size to fully encapsulate the bone. After adjusting the contour around the bone, copy it by selecting an edge of the VOI box and holding the Ctrl and C keys on your keyboard. Next, return to the Slice Position box and load an ending slice. Paste the contour over the bone by holding the Ctrl and V keys on your keyboard. Use the Select tool to adjust the contour if necessary. Select the ending slice as shown at the bottom of the screen to create the slice range. A blue box should form around this range. At the bottom left corner of the screen, select the Morph Range button. An array of pink boxes should form between your selected starting and ending slices. This has pasted your drawn contour for every slice in this range as shown by the red contours. Ensure the contour fits around the entire bone. If not, adjust the VOI box as necessary and morph the range again. Once complete, switch to the Tasks tab and navigate to the Script drop-down menu. Select script number 11 titled Segmentation of One Phase Object. At the bottom left corner of the screen, replace the Z value with your starting slice and your DZ value with the number of slices it would take to reach your ending slice. In other words, the DZ value represents how many slices are to be analyzed from the Z value. Next, ensure the per mile dropdown has been switched to density. Before starting this task, it is strongly encouraged to adjust the lower threshold value to create a better 3D image. Once complete, navigate to the top left of the window, click File, Save Contour As, and then save the contour as the default GovJ file. We are now ready to run the task. Click the Start Task button, and then click OK. Use the task history window to view the job queue and the status of the segmentation task. If there are additional bones that require this process, you may load those measurements and repeat these steps. Once the script has finished running, you may close out of the entire application and load the ray tracer application. Before locating a measurement, ensure the segmented bubble is selected. Once an image has been loaded after clicking the Start button and any pertinent changes have been made, we can now export it. At the top left corner, click File, Print, TIFF, and then scratch temp. The file name may be adjusted if you wish to do so. It is extremely important to record or memorize this file directory before saving the 3D image to locate and access it later on. Finally, click OK and feel free to either close out of the ray tracer window or load another 3D image to export. To access your 3D images, click on the File Browser icon in the bottom left corner of the screen. Follow the directory that was shown in the ray tracer application when saving the 3D image. Once you reach the SCANCO user folder, locate your TIFF file via the name and the date modified. You may open this image to ensure it is yours before using methods such as Box to export it.